Hi everyone. Let me discuss about overview of immunology. So first of all, the introduction. The immune system is a remarkably versatile defense system that has evolved to protect animals from invading pathogenic microorganisms and cancer. It is able to generate an enormous variety of cells and molecules capable of specially recognizing and eliminating an apparently limitless variety of foreign invaders. These cells and molecules act together in a dynamic network whose complexity reveals the depth the nervous system. So immunology grew out of the observation that individuals who had recovered from certain infectious disease were thereafter protected from the disease. The Latin terms immunis mean exempt, or exempt, I mean the state of protection from infectious disease. So the phenomenon of immunology, immunity, immunis, so they're given by Thucydides. A plague in Athens, period of 430 BC, that only those who had recovered from the plague could nurse the sick because they would not contract this disease a second time. Also, this immunity was given by Thucydides. So this phenomenon, the historical important phenomenon also given by Thucydides. Early society recognized the phenomenon of immunity almost 2000 years post before the concept was successfully converted into medically effective practice. The first recorded attempts were performed by the Chinese and Turks in the 15th century. In 1780, Lady Mary Orclay Montagu observed the positive effect of variolation on the native population and had the technique performed on her own children. The method was significantly improved by English physician Edward Jenner in 1798. So Lady Mary Wortley Montagu performed this technique on their own children. So the techniques also named is variolation. So the that is a tried crust. The effect of poison also have the tried crust. So the tried crust are collecting from the effect of poison. So the tried crust also uh, inhaled through nostril or the incision through small cut opening on their skin. So then after that poison also protected from the same disease. So this technique also known as variolation. Then intrigued by fact that milkmaid who had contracted the mild disease of cowpox were subsequently immune to smallpox. Jenner reasoned that introducing fleet from a cowpox pustules into people, inoculating, for example, inoculating them there, the cowpox pustules also taken from the pox pustules. Then after that, the substance was successfully transferred to the healthy person. So the healthy person might protect the from smallpox. So the cowpox pustules also protect the people against smallpox. Louis Pasteur has succeeded in gluing the bacterium thought to cause bowel cholera in culture and then had shown that chickens injected with the cultured bacterium developed cholera, proved that aging had weakened and the virulence of the pathogens and that such an attenuated strains might be administered to protect against the disease. Vaccine, the Louis Pasteur also coined the term vaccine. So vaccine are from the Latin word vacco. Vacco means cow. So who is going to honor the general work with cowpox inoculation. Pasteur first vaccinated one group of sheep with heat attenuated anthrax bacillus. He then challenged the vaccinated sheep and some unvaccinated sheep with a virulent culture of the bacillus. All the vaccinated sheep lived and all the unvaccinated animals died. These experiments marked the beginning of the discipline of immunology. In 1885, Pasteur administered his first vaccine to a human, a young boy who had been bitten repeatedly by a rabid dog. Then another one milestone, Emil von Beijering and Shibasovra Kitasato in 1890 gave the first insights into the mechanism of immunity, earning von Beijering the Nobel Prize in Medicine 1901. So these are the scientists also contribute to the field of immunology. Then in 1930s, mainly through the effort of Elvin Kovac, a fraction of serum first called gamma globulin. In 1883, Mechenkov demonstrated that cells phagocytosis is also contribute to the immune state of an animal. In 1950, lymphocytes T and B cells identified by Bruce Click at Mississippi State University. Early theories attempted to explain the specificity of antibody-antigen interaction. In the beginning of 19th century, Jules Baudet at the Pasteur Institute expanded the concept of immunity by demonstrating specific immune reactivity to non-pathogenic substances such as red blood cells from other species, called Landsteiner blood grouping. So in the beginning of 19th century, Carl Landsteiner also discovered the blood grouping antigens. Then Emil Fischer in 1894 to explain the interaction between an enzyme and its substrate. Then Paul Ehrlich proposed that the beginning of receptor to an infectious agent was like the fit between a lock and key method. The immunity, the state of protection from infectious disease, both a less specific and more specific components. The less specific components, innate immunity, provide the first line of defense against infection. Immunity, innate immunity are present before onset of infection and constitute a set of disease resistance mechanisms that are not specific to a particular pathogen. The specific components, adaptive immunity, does not come into play until there is an antigenic challenge to the organisms. Respond to the challenge with a high degree of specificity as well as the remarkable property of memory. Adaptive immunity or lymphocytes and the antibodies and the other molecules they produce. Types of immunity. Innate immunity provide the first line defense most of the microorganisms encountered by healthy individuals. Immune system is to prevent infection by pathogenic microorganisms like bacteria, viruses, parasites, fungi, etc. The body defense mechanism can be divided into two types, innate and natural immunity. Second one, acquired or adaptive immunity. Innate immunity provides the first line defense against infection. Cellular and molecular components that recognize the class of molecules peculiar to frequently encountered pathogens. Phagocytic cells such as macrophages, neutrophils, etc. Innate immunity may be classified as individual innate immunity, racial immunity, and species immunity. Individual immunity resistant to infection which vary with different individuals in the same race and same species. Racial immunity difference in resistant to infection among different species within a same species. Species immunity total or relative resistance to a pathogen shown by all members of a particular community. Is it clear? Okay. Then factor affecting innate immunity. There are many factors can affect the innate immunity: age, nutritional status, the hormonal levels, etc. Age. Extreme of age make an individual highly susceptible to various infections. Nutritional status. Both humoral and cell-mediated immunities are lowered in malnutrition. Hormonal levels. Individuals with certain hormonal disorders become increasingly susceptible to infection. For example, diabetes mellitus, hypothyroidism, adrenal dysfunctions are increasingly susceptible to infection. Mechanism of immunity. The innate immunity of the host performs two most important functions. The first one, it kills invading microorganisms. Second one, it activates acquired immune response. The first of all, the immune system try to kill invading pathogens. Second one, your immune system also activate our immunological process. Innate immunity is primarily depend on four types of barrier: anatomical barrier, physiological barrier, phagocytic barrier, and inflammatory barrier. These are the four barriers which will be involved in innate immunity. Atomic barriers to our skin and surface of mucous membranes are effective barrier. The skin consists of two distinct layers. The thinner outer layer known as epidermis. It consists of dead cells and is filled with a water-proofing protein called keratin. A thicker layer called as dermis, connective tissue, contains blood vessels, hair follicles, sebaceous glands, and sweat gland. Sebaceous gland. Sebaceous gland can secrete sebum, the pH range 3 to 5. Physiological barrier, temperature, pH, variable soluble proteins also known as physiological barrier. Normal body temperature inhibits the growth of pathogens. pH, gastric acidity, low pH of the stomach content. Various soluble proteins, lysozyme, interferon, complement also known as variable soluble protein. Phagocytic barrier. So the phagocytic cells also act as a barrier. So which can be able to swallow that newly invaded microbes. The newly invaded microbes also digested by phagocytic cells because phagocytic cells also contain phagosomes. The phagosomes also known as the back which will be contain lytic enzymes. The lytic enzymes can be able to degrade that bacterial, the swallowed bacterial cell. So the finally the digested bacterial cells also released from the macrophage. Inflammation barriers. So the tissue damage caused by a wound or by an invading pathogenic microorganism induce a complex sequence of events collectively known as the inflammatory response. The sign of inflammation is rubor, tumor, calor, dolor, then functional lesia, loss of function. So thank you for watching this video. Thank you.